Hello, everyone. Welcome to today's live broadcast, Keeping Up with New Emerging Analytical Technology. I'm Shannon Stoltz, Custom Content Specialist for Biopharm International, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by Biopharm International and sponsored by 908 Devices. Now, before we start, we do have a few quick announcements, the first being that this webcast is designed to be interactive, so we encourage you to ask questions throughout the event. You can submit any questions you have by typing them into the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player, but please keep in mind that the slides will be advancing automatically during today's event. If you have any technical problems either viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. Moving on, I'd like to introduce our great speakers for today. First, Nicole Azer began working with the U.S. Food and Drug Administration in the Office of Biotechnology Products in 2019 as a fellow with the Oak Ridge Institute of Science and Education. She works in the Bioprocessing Lab and Office of the Pharmaceutical Quality Research to explore process analytical technology, evaluate process changes, and understand potential product quality impacts in mammalian expression systems. She holds a master's degree in biopharmaceutical engineering from Keck Graduate Institute. And then Graziella Piras leads the strategic marketing activities for the Life Sciences Division at 908 Devices. And she has over 18 years of experience developing applications and solutions for upstream bioprocessing. Grazia previously held various R&D and marketing positions at Thermo Fisher Scientific, where she led projects to support the cell culture and cell therapy business. Graziella received her PhD in biochemistry and molecular biology from the University of Liège in Belgium. She did her postdoc studying the role of gene expression and epigenetic regulation on cancer and development at the National Cancer Institutes in Maryland. So thank you both so much for joining us today, and please get us started.
Thank you so much, Shannon, for the very kind introduction. I'm Graziella Piras, Senior Director of Strategic Marketing at 908 Devices, and I am very happy today to be introducing the webinar, Keeping Up with New Emerging Analytical Technologies. So it's been 20 years since the FDA released the Framework of Process Analytical Technologies, or PAT, which encourages drug developers to adopt innovative technologies to better monitor and eventually control the bioprocess. So in the upstream part of the process in the bioreactor, this could entail real-time monitoring with inline and online devices that measure key analytes in the bioreactor directly. But it could also entail at-line devices or sometimes even offline, taking samples and analyzing them as close as possible to the bioreactor, and importantly, as frequently as possible to better achieve a better cell culture and eventually a safer and more efficient drug. Many companies have been adopting different forms of PAT technologies, and also many tool developers have been developing the right tools to enable this great approach. And including 908 Devices, we've been developing several tools to enable inline, online, and at-line and offline, also PAT technologies. So where in the process are these PAT technologies used? Eventually in the manufacturing process to better monitor and control the manufacturing process, but also in early and late process development where the PAT approach are being developed, but also help in optimizing, for example, the cell culture media, developing platform median feeds or optimizing median feeds during the later phases of process development, as well as actually optimizing the process, maybe to refining a feed strategy. Being able to monitor and have a feedback control is great when developing feeding strategy, for example, or using PET technologies to develop a model process to have a virtual representation of the process eventually, and being able to do the right experiments. So many places where PET is used, and I'm very happy to hand it over now to Nicole, who's going to talk about how she's been developing and looking at PET technologies for the process she's working on. So Nicole, over to you.
Thank you so much for the lovely introductions, and a good morning. Thank you for joining my talk, and we will be keeping up with the new emerging technologies. I am Nicole. I work at the FDA, and I'm a part of the Office of Pharmaceutical and Quality Research. And of course, this presentation reflects the view of the author, me, and should not be constructed to represent FDA views or policies, and I have no competing financial interest. Of course, at the FDA, we want a quality product of any kind consistently meets the expectation of the user, and drugs are no different. Patients expect safe and effective medicine with every dose they take, and pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. And it is what gives patients confidence in their next dose of medicine. Now, what we're going to be talking about today, I wanted to go through just a little bit of a background, what my research goals are, and with my group, a little bit of background on amino acids, glycosylation, why do we care, and some projects that we work on, how we see media affecting glycans, and further spent media analysis, challenges and method comparisons about how we analyze our spent media, kind of an overview of our samples, as well as our amino acid trends that we've seen, and some conclusions of our latest experiments. So in my lab, I am in the bioprocessing lab, and we are together with upstream and downstream. I'm a part of the upstream where we are able to expand and shake flasks. We have various bioreactor platforms, including the Amber modular systems, stir tanks up to 5 liters, and soon maybe a pilot bioreactor. Of course, we are working with a downstream team, and they focus on viral clearance, continuous chromatography, and we also have some subvisible particle projects as well. Now, my group, we focus on upstream and process analytical technologies, or PAT, because they provide a way to understand how cells in bioprocessing grow and how they produce protein biotherapeutics. Of course, with media and feed studies, we need to look at all our various cell lines that have their very specific feeding requirements in order to produce these very high quality products. And this is typically done with mass spectrometry or MS methods. Of course, all together, along with multiple cell lines, all these feeding strategies, long culture times, and the development it takes for mass spec methods as well as process development, this overall becomes very time consuming and labor intensive. And this is really the challenge that we're trying to look towards supporting. Now, a little background on amino acids. Of course, these are the building blocks of proteins and another potential energy source in cellular metabolism. Some challenges we see with mass spectrometry is that they are all different in side chain chemistries, which lead to ionization efficiency differences, and of course, hydrophobicity differences. And an important thing with amino acids is leading to product quality. They always say you are what you eat. And one of the product qualities that we look at is glycosylation, specifically N-linked glycans. These are oligosaccharides that are attached to the Fc region on a conserved asparagine residue on monoclonal antibodies or mAbs. Glycosylation is a critical quality attribute, and it can affect therapeutic performance. We've seen that it affects clearance rate, efficacy, and then, of course, immunogenicity. Now, what we're really looking at is, does amino acid affect mAb glycosylation? We've seen that certain forms of glycoforms have different effects on mAbs, and we see that high mannose glycoforms can have faster clearance rates and therefore reduce effective half-life. Further, fucose glycoforms can inhibit antibody-dependent cell-mediated cytotoxicity and therefore decreases the efficacy. And finally, we've seen that alpha-galactose glycoforms can be immunogenic, and this really compels processes to minimize this production. Some of the previous projects we've worked on in my group is studying how media can affect glycan profile, where we looked at several different types of medias, and we were able to see that there are indeed different glycan profiles depending on what media we used. Now we wanted to take this a step forward and bring in online amino acid analysis. We were able to do this with a FIA lab, which helped us create an OPC network, and we were able to connect our 5-liter stir tank reactors with an automatic sampler. And FIA Lab's Procea was able to do the automated chemical derivatization of amino acids and then further send it to a LCMS with a single quad mass spec in order to capture real-time and online amino acid analysis. And this way, we wanted to explore how amino acid trends could be linked to glycosylation outcomes in just a monoclonal antibody. To do this, we used multivariate data analysis and principal component analysis to study the association between amino acids and glycan patterns. Principal component analysis can be used in large data sets to simplify visualization and analysis. To sum it up, it's really to reduce the number of variables of a data set while preserving as much information as possible. And we were able to see with our online amino acid analysis, we were able to see that the third and fourth principal component were not as confounded by feeding strategy or batch age like the first two principal components. Now, taking these third and fourth principal components, we were able to use loading weights, which really allows us to visualize these complex associations. And we were able to plot the third and fourth principal component against each other. And the third principal component along the x-axis showed us that there was an association between the G0F, G1F, and G2F with alanine and phenylalanine, which gave us an idea of further studies that we need to look into for other amino acids. Now, when we're looking into the future studies of metabolism and media effects, it can be quite time consuming. And just to map out a little bit of what that would look like in design, when we're evaluating various process changes, we have multiple cell lines with various medias and specific feed strategies. Now, for example, let's say we've got 3 cell lines. You want to have triplicate biological replicates, and you've got your original media, and you've got 2 new medias you wanted to study with. Now, of course, there's the 3 shake flasks you want to have for your original, and then you're going to have a 50-50 with your first new media and your second new media, and then 100% of your first and second media. Now, altogether, this is 15 shake flasks we've got to take care of for probably about 14 days. And let's say we only want to do 1 sample a day. That's already over 200 samples for analysis per cell line, let alone all the back end work of maintaining these cells. You also have to do spent media analysis. Traditionally, spent media analysis is done with LCMS, and this is to identify and quantitate amino acids as well as other metabolites. But LCMS requires a very high level of technical skill. It's time consuming for method development. It can be very labor intensive with data processing and data management. Essentially, everybody wants mass spec data without doing the mass spec work. And PAT has been a tool to really improve this bottleneck and support these challenges in bioprocessing development. So what we wanted to look at is kind of see the new MS ways. And so we see traditional LCMS is a very well characterized method that's been developed over decades. It's got a very flexible application of methods. And we also are now seeing 908 devices with their new capillary electrophoresis MS. And this is specifically designed for cell culture and spent media analysis, but because it's new, it is quite limited in publications to understand its novel technology. So we really wanted to see how we could fit that into our group. And so we were looking at 2 different methods, essentially. We look at our traditional LCMS, which is just a Waters BioAccord RDA. They have a amino acid cell culture standard kit, requires some manual serial dilution preparation. What's nice about this is that it is underivatized and you can analyze neat cell-free spent media. And we were able to use a Waters application note in order to optimize that system for our setup. Now we wanted to look at the CEMS, the Rebel, which has its specific spent media analysis kit. It's already got standards that are pre-prepared, inject ready, and they also have a neat cell-free spent media sample prep, and that also includes underivatized samples. And then, of course, they have a media range finding tool, which really just helps you identify the dilution per media changes. Now, just to give a little bit of an overview for the cell culture runs that we've done, we essentially look at two different cell lines here. We are working with a VRC01. It's a recombinant CHO-K1 expressing model IgG1 monoclonal antibody. And then the other cell line that we are working with is NIS-CHO, another recombinant CHO-K1, and it is expressing our NIS-MAB here. Now, both these cell lines have specific chemically defined medias. The VRC01 has using high clone ActiPro, as well as its own specific 7A, 7B supplements. And similarly, NIS-CHO cell line has its own chemically defined media that we're looking into, as well as additional feeds. Now, for all the bioreactor runs that we've ran and we were analyzing here, we've got our Amber 250 modular bioreactors, and we were able to run VRC01. We did 2 sets of 2 biological replicates. And overall, it was a 10-day culture fed batch, and we were able to get 32 samples from that. Additionally, we ran our Amber 15, their modular bioreactors, and we had 2 sets of 6 biological replicates, a total of 12 samples that we harvested. This was a 9-day fed batch culture. And then finally, for our NIS-CHO, we ran our stir tank 5 liters in 3 biological replicates. This was about a 6-day culture in batch mode. We got 36 samples. And then finally, Amber 250, where we did 4 biological replicates of our NIS-CHO with a total of 32 samples over a 14-day culture in fed batch mode. So once we ran all those and we had all our samples, it was time to do our spent media analysis. Now, the Rebel has a media range finding tool, which is essentially a calibration curve analysis. It's an automated tool, and it is a way for you to determine what the best sample dilutions will give you the best coverage or whatever you're looking for. So this is specific to the standards included with the kit. And here I'm showing an example from our XL Advanced, which was for the NIS-CHO cell line in their fed batch medium. And we chose 100x because we wanted to get we wanted to get as much as we could, and really just taking out of those 3 that we weren't able to see, but that way we were able to get most amino acids that we wanted to look at. So now, using the media range finding tool in practice, it was an example you can see here is that we're looking at valine, and it's smack dab right in the middle of a calibration curve, as you can see in the green line. And that's really where you're kind of looking for that sweet spot. And in both these batches, you can see that we've got a really nice trend line where we see the red line is the LCMS BioAccord RDA, and the blue line is the Rebel CMS. And we were able to see, like with valine, we were able to see these nice trend patterns working really well. And then additionally, we were able to take this a step forward, and you can see in asparagine where it's a little bit lower on that calibration curve. We're looking kind of towards the end, not right in the middle where we're usually really looking for, but you're able to really consistently see that, you know, the Rebel does have a lower detection limit, and it was very consistent across all of our replicates and a couple different batches where that fell off. Now, when you're looking at 2 different medias, that's really where you have to pay attention to your calibration curve, because here we see that proline for XL Advanced media, it's right in the middle where you want it, but then in our high clone ActiPro, it's a little bit lower on that calibration curve. And we were able to see that while we're getting good trending while it's right in the middle, you can kind of see, oh, in the high clone, we're possibly at a saturation level, and we were already told that with that media range finding tool. Being a little bit lower, if we wanted to look into proline more specifically, we would likely need to do a lower dilution. So overall, we wanted to see, does Rebel CEMS and LCMS generate similar data? And in order to do this, we wanted to do a statistical analysis with a t-test and a paired t-test. So overall, we have 112 total samples for all 20 amino acids. And the point is to compare the means of 2 groups and determine if these 2 groups are different from one another. Of course, with our harvested cell culture fluid samples, we analyze this with the t-test. But with our other samples where it was a time course, you know, samples were taken throughout the culture and they were analyzed with a paired t-test. And we start this analysis with the null hypothesis that measurements made with the rebel CEMS are different than measurements made by the reference LCMS. Then we look at our data and we look for data-based evidence that these measurements are the same. Now, there are a couple of tricky amino acids in analysis. We are still in progress with the statistical analysis on all the reactor samples and looking at the relative error of the CEMS and LCMS, and then also comparing the amino acid trends, as that's really important for cell culture, is the overall trends and consumption patterns of amino acids. Cysteine is, of course, difficult to analyze due to its solubility. Standards are typically in the form of cysteine, and so the calibration curves are half the concentration. So you're limited a little bit more in your calibration curve for that. Of course, glutamine and glutamic acid are difficult to differentiate. Even in LCMS, it requires some optimization because there is only a mass difference of one. It's really the difference between a nitrogen and an oxygen. But 908 Devices does report a term GLX, and this is when glutamic acid and glutamine are potentially co-eluted in the results, and so that's already upfront. Essentially, in conclusion, we are able to use a lot of PAT that supports a lot of these challenges for efficient process development. I mean, this emerging technology gives us more time to really focus on cell culture process development and impacts of process changes. Some projects that we're looking at is cell substrate impacts on product quality, or other media studies that optimize feeding strategies. And then, of course, metabolic studies for flux balance analysis. Now, a lot of this just becomes a really, really large sample set, and having PAT that makes that more manageable is really the key here, because with high throughput sample testing, easy to use methods for analysis, it simplifies data processing and gets you those faster results so that you can move further along in your experiments faster. Of course, I have to acknowledge my whole team. This is a lovely picture we took at Crab Day and eating those delicious Maryland blue crabs. And without them, none of this would have been possible. So thank you all. And thank you for attending my talk. I will pass it back to Shannon for questions.
Fantastic. Thank you so much for that great presentation. Now, before we jump into the question and answer session, I would like to quickly remind our audience how to submit questions. You can submit questions by typing them into the Q&A box, which can be found directly below the video player. Our first question today is, how can this type of spent media analysis be helpful in the development of biosimilars?
Hi. Yeah, I thought that this was a really interesting question. I mean, this is something that we're working on in our group a lot and focusing on biosimilars. Really, I think that the efficiency of time and being able to have that high throughput sample testing is really, really beneficial. And also, kind of what we're looking at right with NIST-CHO is essentially kind of gauged towards answering those questions of biosimilars, right? Kind of seeing, right, you are what you eat. So if you do change, either cell substrate or maybe your media, depending on what your process changes are, how much does that really affect your product, and are they clinically significant? And I think that a lot of that, by understanding the roots of it, like what you're feeding and going into the products, I think that's a really good basis for understanding biosimilars and fully understanding how they're developed and how those changes in development, if they're clinically significant or not. It just really builds up that foundation for us. So I think that's really interesting.
Fantastic. Thank you very much. Moving on to our next question, what are other real-time CPPs that can be combined with SMA data to provide additional insights into process development?
So mainly we look at, I talked about N-glycans because that's the biggest, CQA that we look at. I know that another one that we're always interested in is charge variants, and seeing how that changes throughout a process. Now, putting that in real time, I think that's going to be something that we can look forward to in the future. I think right now trying to get something online for, either online amino acid analysis in conjunction with online glycan data. I mean, that's really the gold pot of the end of the rainbow that we're all kind of shooting for. But my guess would be charge variants would be an interesting one to look at.
Wonderful, thank you. And then what are the main bottlenecks of traditional SMA?
With traditional like LCMS, I mean, really, it really is just time method development. I think that, yeah, I did spend a lot more time, I would say, developing an LCMS method than, of course, like the benefit with the Rebel 908 devices is that it is kind of like all prepackaged in kits. I mean, that's kind of the era that we're in. I mean, it is, of course, like specifically for cell culture media analysis. That's what it's designed for, at least. But, you know, you do kind of sacrifice that like flexibility, right, that you would get with an LCMS. But I think that that kind of time and high technical experience, you know, that's going to be kind of hard and not as like ubiquitous across like other companies if they're looking for that development.
Thank you very much. And then what was your overall experience with these emerging technologies?
I mean, I'm not going to say it's all happy sunshines. I mean, it's really fun working with a lot of them, but of course with new technology, there comes like a lot of nuances in understanding how they work together. I think one of the biggest challenges is really just getting all these pieces of different technology from different companies working together. Because I think that right now we're really in a collaboration focus of, you know, making these things actually happen. Like we talk about, I've been talking about like online and real-time data, but, you know, that is, you know, takes multiple different devices from different companies in order to make it working together. So like trying to get that harmony almost across all the different companies is sometimes a bit of a challenge, but it is very exciting kind of being right on like that edge of technology and learning and kind of getting ahead of what the challenges might be, so that way we can kind of look at studies that are more, what's the word I'm looking for? I guess preemptively answering a question rather than being reactive towards a problem and popping up. You know, if you can spend more time ahead of it, then you can kind of gauge where there might be some troubles in answering those solutions.
Perfect, thank you. Moving on to our next audience question, can you comment on the actual time savings you experienced using this type of technology?
I mean, I did save, I would say, like a few months. I did have a funky setup, I will say, for my LCMS, just with the way I had it designed in my lab at that time. So it did take me like probably a few extra months to make sure I had like a good cal curve and everything set up, versus, I mean, it was already pre-designed in kit with the 908 devices Rebel. So that was really just training and then taking some time in, I guess, looking back at like the dilutions and the cal curves. So I think it did save quite a bit of time for me, at least. I spent a lot more time fussing with my LCMS, that's for sure.
Great, thank you. And then this Rebel technique is compatible with only Waters MS or any TOF or Orbitrap can be attached.
I'm not sure if this is more, if you mean like you're trying to use the Rebel with another device. I only have the Waters mass spec that I'm working with, so that's just what I'm looking at for my LCMS. So I'm not really sure if the question's targeted for if you can use the Rebel attached to an Orbitrap, because I think I've heard of that. I don't know, maybe Graziella can speak more to that.
I can chime in here. Yeah, so the Rebel is a standalone device. You don't need to attach it to anything. It has an incorporated separation and mass spec detector, so it stands on its own. You don't need to attach it to anything. Put your samples in, and you get concentrations of your amino acids in your media or spent media samples. So it is a standalone product that doesn't need anything. Nicole showed a comparison with a more traditional method, but it stands alone. It does its own thing.
Wonderful, thank you. And we also have a question asking about a copy of the slides. I know this will be available for replay on demand, but I don't know if you have any more information on additional materials.
We did post some information on the platform about the Rebel. There's a lot of information on the 908devices.com. platform, website. I mean, so, and yeah, and we can always reach out to the specific person asking for the slides.
Perfect, thank you. And then our next question, can you use it as well for amino acids in downstream processing?
So that's an interesting question. In theory, you know, any sample can be analyzed. It's going to be a matter of, we have not tested that ourselves. The Rebel was designed, like Nicole very nicely showed, for spent media analysis in upstream. So to be determined, to see, the question is, are the amino acids in concentrations that the Rebel can detect? But in theory, you could analyze samples from different things.
All right, wonderful. Thank you very much. And with that, it does look like it's already time to wrap up. So I want to thank the audience for attending and participating in today's event. And I would also like to thank our sponsor, 908 Devices, for making today's educational webcast possible. We would also like to ask everyone in the audience to participate in a brief survey, and that will be appearing on your screen after today's presentation has ended. And as I noted, you'll be receiving an email alerting you when this webcast will be available for replay on demand, and we invite you to forward that announcement to your colleagues who may have missed today's live event. We hope to see everyone next time. Thank you and take care.
Thank you.